Hello guys and welcome back to Accenture Academy. So this is another numerical from the mole bio. It is based on the restriction sites only. So let's do our question today. So the question says the restriction site for an enzyme 191L is 5-CCA NNNNNTGG. How many times can this enzyme can cut a 4 MB bacterial genome given that 50% GC content is present? Okay. Now, this looks very twisted, but it is not. So first of all, as we have discussed in the previous numerical section also, you need to calculate the probability of each of these bases occurring. Okay. So you need to have this sequence first. So let's write the sequence first. So it is 5 dash C C A N N N. Then there is a cut N T G G 3 dash. Okay. Now this cut means that the enzyme is going to make a cut over here. Okay. And N means any bases can occur. So it can be A, T, G or C. Now we need to calculate the probability. So we know that again we have got the 4. So the probability is going to be each of these is going to have 1 upon 4th. Okay. So now I'm going to calculate the probability of each of these. So it is given as for C again 1 upon 4th, C 1 upon 4th, A 1 upon 4th. Now N is given. So N means what? Any base. So the probability of occurring A, T, G, C is going to be equal, right? So it can be, so we can say that all the bases can occur. So that's why either you can say that there is no probability or you can write it as 1. So 1, 1, 1, 1, 1. So we have got 5 times 1 is there. Okay. So anyways, it is not going to make a difference because 1 into, sorry, it's not 5, it's 1 raised to 5. Okay. Yeah. So it is not going to make a difference because you know that anything multiplied with 1 is going to give you 1 only. Now coming to T, it is 1 upon 4th, G 1 upon 4th, again G 1 upon 4th. Now what is the total we got here? So we have got 1, 2, 3, 4, 5, 6. Okay. So that becomes 1 upon 4th raised to 6 and we know that it is going to be 1 upon 4096. Okay. So we have done so many times. So when you keep on calculating, you know, all these questions, so you tend to remember them. So now we know that 4 raised to 6, you will get 4096. Okay. So you can remember now. So it becomes easy for you guys to clear during your exam. If you can remember all these. Okay. Now it is also given that it is also given that, so you have got the 4096, okay, and it is given it has got the 4 MB bacterial genome with 50% GC content. So when it is given that 50% GC content, it means what? 25% G, 25% C. So with this, we know that 50% is going to be your AT content. So again, you will have 25% A and 25% T. So since all this percentage is going to be same, so we don't have to include this. So it is going to be same. So we don't have to do any changes in the question. Okay. So just let me have a clear space. Now it is given that, so we had 4096 base pair and it is given as 4 MB bacterial genome. Now how can you write 4 MB? What do you mean by 4 MB? It is 4 into 1000 into 1000. Okay. 4, this becomes kilo, this becomes your mega. Okay. Now if it is, so if it is 6, it is mega. If it is 10 raised to 9, it becomes giga. So if you, please remember all these things. Okay. So you have got 10 raised to 3 means kilo. This is mega. This is giga. So please remember all these raised to figures also. So this is your 4 MB bacterial genome. And now we know that after each 4096 base pair, this enzyme is going to make a 1 cut. So how many cuts are there? So 4 MB divided by 4096. So when you calculate, if you can take it, so what you get? You can just get a approximation of around 1000 times. Right? So you don't have to get the real accurate answer. You can make this 4096 as, you know, near to 4,000. So what you can get? Cancel, cancel, you get 1,000. So near about 1,000 times I'm getting. So this is your answer. So this is how you're going to calculate, since it is the approximation, unless until they're asking the specific digits. You don't have to calculate, okay? You can just go with the approximation. So this is going to save your time also. So I hope you guys understood all those things. Still, if any doubt is there, please you can comment me in the comment section, or you can also mail me at excelsioracademy1228@gmail.com. There's a Telegram channel also by the same name, Excelsior Academy. You can join there and ask any questions. Guys, you can also put your questions in the comment section so that I can make a solution video for those. So thank you guys for listening. Keep excelling with Excelsior Academy. Thank you guys.